Chapter 3.4, The Cell Interior. Read-along mode. Follow along, no excuses. All right, Jake, we're straight into 3.4. This is where we rip open the cell and see what's actually running the show inside. No weak-ass surface-level bullshit. It's still surface level is bullshit. We're breaking down each organelle, calling out every bold term, and making sure you own this knowledge. If you're holding the book, track with me. Every time you see bolded words, make sure you can point to them on the page. Every time we hit an image, I'll break it down so you know exactly what's going on. 1. The cytoplasm, the work zone. First paragraph. Follow along. As soon as we get past the plasma membrane, we're in the cytoplasm. This is where the real work happens. Cytoplasm is everything inside the cell except the nucleus. Cytosol, ICF, the fluid portion of the cytoplasm. This is where the organelles float around and chemical reactions happen. You screw up your hydration, cytosol gets messed up. Cells don't function. You start feeling sluggish. Image breakdown. The diagram shows organelles floating in cytosol, surrounded by the plasma membrane. You should be able to spot the nucleus, ER, mitochondria, and Golgi. Get familiar with the layout because this is the powerhouse of all cellular function. 2. Organelles, the cell's machinery. New section, stay with me. Each organelle is a specialized unit. Like departments in a factory, you wouldn't expect the HR department to run security, and you sure as hell wouldn't want a muscle cell acting like a nerve cell. The book gives a list. Go through it as we break these down. The nucleus, the control center. Find this paragraph. It's critical. The nucleus is the brain of the cell. This is where your DNA is stored, your genetic instructions for building proteins. Nuclear envelope, double membrane that surrounds the nucleus, keeping the DNA protected. Nuclear pores, tiny holes in the membrane that control what exits the nucleus. Nucleoplasm, the fluid inside the nucleus containing chromatin, DNA plus proteins. Nucleolus, the birthplace of ribosomes. If you don't have this, you can't make proteins and your cells die. Image breakdown. You should see a big round structure labeled nucleus with tiny pores in its envelope and a darker nucleolus inside. Ribosomes, the protein factories. Find this section. It's short but crucial. Ribosomes, where proteins get built. No ribosomes, no proteins, dead cells. Some float freely in the cytoplasm, make proteins for use inside the cell. Some attached to the rough ER make proteins for transport out of the cell. Image breakdown. Find the tiny dots labeled ribosomes. These are literally what let you grow muscle, repair tissue, and make enzymes. Endoplasmic reticulum, the cell's factory. Next section, follow along. Rough ER covered in ribosomes. Its job? Assemble proteins. Smooth ER, SCR, no ribosomes. It detoxifies, builds lipids, and processes steroids. No smooth ER, you can't detox shit. Liver cells rely on this. Alcoholics destroy their SER. Image breakdown. Look for a network of tubes. RAR will have dots ribosomes. CR is smooth. Golgi complex, the shipping center. Find this paragraph, super important. Golgi complex processes, packages, and ships proteins to where they need to go. It modifies proteins before they get sent to their final destination. It creates vesicles for transport, including lysosomes. Image breakdown. Find the stacked, pancake-looking organelle. This is where proteins get their final quality check before being shipped out. Lysosomes. The cell's garbage disposal, short but powerful section. Lysosome. Chapter 3.4, The Cell Interior, continuing from lysomes. Read along, Moke. No excuses. End of this, you better have a clear-ass picture of what's happening inside your cells. Let's keep rolling. Peroxisomes, the detoxer. Find this paragraph, next after lysosomes. You thought lysosomes were the only cleanup crew? Think again. Peroxisomes are like your chemical waste disposal system. They break down toxic molecules before they can kill the cell. Peroxisomes contain enzymes that detoxify harmful substances, alcohol, free radicals, and toxins, breaks down fatty acids for ATP production, produces hydrogen peroxide, H2O2, as a byproduct, then breaks it down before it becomes toxic. Without peroxisomes, your liver and kidneys would shut down, and toxins would fry your cells from the inside. Image breakdown. Small, round vesicles, similar to lysosomes, but specifically handling detox and fat metabolism. Mitochondria, the powerhouse of the cell. Find this section. You know this one. All right, this one's famous as hell. But if you only remember the phrase powerhouse of the cell, you're a goddamn amateur. You need to know how and why mitochondria matter. Mitochondria make ATP, adenosine triphosphate, the only energy your cells can use. They have a double membrane. Inner folds called cristae increase surface area for ATP production. They contain their own DNA, mtDNA. Mitochondria were once independent bacteria. More mitochondria, more energy. Muscle and heart cells have tons of them. If mitochondria fail, you're done. No ATP, no muscle contraction, no nerve impulses, no cell function. Image breakdown. Look for a bean-shaped organelle with folds inside. Those folds, cristae, are where ATP gets made. Centrioles, the cell division commanders. Find this section. It's small but powerful. When cells need to divide, they don't just randomly split apart like some dumbass crash test dummy. They use centrioles to organize the entire process. Centrioles are two perpendicular cylinders made of microtubules. They form the mitotic spindle, which pulls chromosomes apart during cell division. They also create cilia and flagella. More on that next. No centrioles? Your cells can't divide properly, and growth and repair go straight to hell. Chapter 3.4, The Cell Interior, Cell Interome. Read along mode. Follow the book, no excuses.